Imagine you're building a super smart artificial intelligence.
Right.
You pour millions of dollars, thousands of computing hours, and terabytes of data into training it. The goal is to perfectly match cancer patients with the exact drug that will, save their lives.
The holy grail of medicine, really.
Exactly. So you give the algorithm its final evaluation and it scores off the charts. It looks like a total medical revolution.
And there's always but you dig a little deeper into the data.
Yeah, and you realize something horrifying. The AI didn't actually learn the complex biology of cancer. It just, figured out how to cheat on the test by memorizing the grading rubric.
It's a pretty alarming realization.
It really is. Welcome to the Deep Dive. Today we are looking at a May 2026 paper published in Nature Communications that exposes a massive, dangerous gap between the hype of medical AI and the clinical reality.
Yeah, we'll be unpacking a new evaluation framework called DrEval. It's basically systematically dismantling how we predict cancer drug responses.
So to really understand why this paper is causing such a paradigm shift in the computational oncology community, we kind of have to look at the baseline goal of the entire field, right?
We do. We have to start with true personalized medicine. We're talking about taking a patient's tumor, mapping its precise biological profile, and using an algorithm to predict exactly which compound will eradicate it.
Because the goal is to skip that awful trial and error phase of chemotherapy, right? The one that costs patients so much valuable time and physical toll.
Precisely. And to train these predictive algorithms, developers are relying really heavily on cell line screens.
Okay, cell line screens. These are massive public databases, right? With acronyms like CCLE, CTRPV2, or GDSC.
Right, yeah. In those databases, researchers are essentially cultivating immortalized cancer cells in a lab. They hit those cells with hundreds of chemical compounds to see what sticks.
And we're ultimately looking at dose-response metrics to measure the efficacy of those compounds.
Yeah, we are. If you picture a graph showing the concentration of a drug on the x-axis and the survival of the cancer cells on the y-axis, you're looking for an S-curve.
And how do we actually, summarize that curve?
Well, we use specific metrics. The primary one is the IC50.
Which stands for the half maximal inhibitory concentration.
Exactly. It tells you the exact dosage required to kill 50% of the cancer cells.
Got it. And there's another word too, right?
Yeah, we also use AUC, or area under the curve. That one summarizes the overall potency of the drug across all tested doses.
So, it sounds like the perfect sandbox for deep learning. You have a detailed biological input, a chemical structure, and a clear quantitative output to predict.
It sounds perfect, but this Nature Communications paper identifies 6 major roadblocks that are just, completely stalling the transition from these lab-based databases to actual clinical success.
Okay, let's unpack this. The first major hurdle the paper tackles is the illusion of big data itself. Let's look at the CTRPV2 dataset.
Right, so that dataset contains roughly 400,000 drug response curves.
Which, for a machine learning engineer, 400,000 data points looks like a total gold mine.
Oh, absolutely. You'd think that's more than enough data to train a neural network with millions of parameters without overfitting.
But you have to look at the actual composition of those 400,000 curves, don't you?
You do. They're derived from only 886 unique cell lines and 545 drugs.
Wait, only 886 unique cell lines? That totally changes the math.
It really does.
It makes me think of a restaurant boasting about having 400,000 5-star Yelp reviews. You assume it's universally loved, but when you scroll through, it's just the exact same 886 people rating the exact same 545 dishes over and over again.
That is a great analogy. You don't actually have 400,000 independent opinions on the menu.
Right. So what do we call that in statistics?
In statistics, we call that pseudo-replication. You are treating those repeated Yelpers as if they were brand new customers.
And I assume AI doesn't handle that well.
Not at all. When you feed highly repetitive, non-independent data into a massive deep learning model, the algorithm's backpropagation doesn't learn generalizable biological rules.
It just wildly overfits.
Exactly. It adjusts its internal weights to memorize the specific quirks of those 886 cell lines, rather than learning the actual underlying mechanism of why a tumor responds to a chemical.
So the effective sample size is a mere fraction of what developers assume it is. But if the models are overfitting to such a small effective sample, how are they still passing the industry's evaluation benchmarks with flying colors?
Through data leakage. And that stems directly from flawed testing splits.
Okay, break that down for us.
Sure. The paper breaks down 4 distinct ways you can partition your data into a training set for the AI to learn from and a testing set to evaluate it. The distinction is crucial for what you're actually claiming your AI can do.
And what's the most common one they use?
The most common, and frankly the most problematic, is LPO leave pairs out.
That's where the researchers just randomly hold back a percentage of drug cell combinations for the final test, right?
Exactly. But think about it. If you use LPO, the model has likely already seen that specific cell line during training. It was just paired with a different drug.
Oh, I see. Or it's seen that drug, just paired with a different cell line.
Right. The model is essentially peeking at the answers. LPO is only valid if your mathematical goal is matrix completion.
Meaning you're just trying to impute missing values in your existing data set, not predict for a new patient.
Yes, but researchers are using LPO splits and then marketing their models as being ready for personalized medicine.
Which is a fundamental scientific mismatch.
Huge mismatch. If you want to claim your AI can predict a treatment for a newly admitted hospital patient, you must use LCO leave cell line out.
So the test set has to consist entirely of cell lines the model has never encountered.
Exactly. And if your focus is drug repurposing, say testing a known breast cancer compound on a lung tumor, you need LTO, leave tissue out.
So the model has to generalize to a totally new tissue of origin.
Right. And what if we're trying to use AI to design a brand new pharmacological compound?
Oh, then you use LDO, leave drug out, right. The algorithm has to predict the efficacy of a chemical structure it has never analyzed before.
You got it.
Okay, we're about 5 minutes in, so let's do a quick recap. Bullet summary time for you listening. Number 1, multi-omics is the comprehensive molecular profile of a tumor. Number 2, pseudoreplication is artificially inflating our AI datasets.
Yeah, making 886 cell lines look like 400,000 independent tests.
Exactly. And bullet 3, data leakage is happening because researchers use LPO splits instead of the much harder LCO or LDO splits.
Which artificially boosts their grades.
Right. So think about your own field for a second. Have you ever seen a metric artificially inflated just because the testing environment didn't match the real world? It happens everywhere.
It really does. And this raises an important question. If the test sets are flawed, what exactly are these complex AIs learning?
If they aren't learning the deep biological mechanisms, what are they doing?
Well, they're likely falling victim to Simpson's paradox. And this is where the findings get deeply uncomfortable for the field.
I know Simpson's paradox usually involves statistical trends that appear in distinct groups of data, but then vanish or reverse when those groups are combined due to a confounding variable. How is that manifesting in cancer drug prediction?
It basically comes down to the vast variance in baseline drug potency.
Okay, how so?
Remember we talked about measuring the IC50 dose? Well, some drugs are highly toxic by nature. They will obliterate cells at a microgram dose.
Right.
Others are much milder and require a full milligram to have any inhibitory effect.
Sure, just like how one shot of espresso drastically spikes your heart rate, but you'd need to drink a gallon of green tea to get the same caffeine jolt. The baseline potency is entirely different.
That's exactly it. Because of that massive variance in baseline potency, the deep learning models discover a mathematical shortcut to lower their error rate.
A shortcut.
Yeah. They realize they don't need to analyze the tumor's complex biological profile at all. They just memorize the average historical effectiveness of each drug across the entire training data set.
Oh, wow. It's the lazy teacher grading essays.
How do you mean?
Like, instead of actually reading the nuanced arguments in your paper, the teacher knows the historical class average is a B-. So whether you spent 40 hours researching the topic or wrote it on the bus ride to school, everyone gets a B-.
That is exactly what's happening. The AI is completely ignoring the individual multi-omic profile and just defaulting to the global mean.
That is wild. How did the researchers prove this?
To prove this was happening, the DrEval researchers constructed what they call naive predictors.
Naive predictors, what are those?
These are baseline mathematical models built entirely on statistical averages. The naive drug mean predictor just guesses the average drug response.
That's simple enough.
And the naive mean effects predictor uses a simple formula. It adds the average historical effectiveness of the drug to the average baseline vulnerability of the cell line.
Wait, are these naive predictors using the multi-omic data at all? Are they looking at the gene expressions or mutations?
Zero biological input. Seriously. They do not look at a single gene or a single molecule. They just calculate the statistical means. And here is the major revelation.
Let's hear it.
When they ran these zero biology models in the LDO setting leave drug out, where the model has to evaluate a totally unseen drug-no complex neural network, could beat the naive mean effects predictor.
That is a staggering reality check. We are throwing massive computing clusters, millions of dollars, and incredibly dense genetic data at these neural networks.
Yep.
And when faced with a new compound, they perform no better than a basic calculator just guessing the historical average.
It actually goes a step further.
Yeah.
The DrEval team took the advanced AI predictions and applied normalized metrics.
Normalized metrics, what does that do?
They mathematically subtracted the naive mean predictions from the AI's results. They stripped away the teacher, giving everyone a B- to isolate the actual biological insight the AI had supposedly learned above and beyond the average.
Let me guess, the performance flatlined.
It completely fell off a cliff. Once the models couldn't rely on the crutch of memorizing the average drug response, their predictive power evaporated.
Wow.
Yeah, it proved that the complex architecture was effectively a mirage. They were capturing almost no generalizable biomedical insights.
Okay, time for our second recap. Bullet 1. Simpson's paradox means AIs are memorizing baseline drug potency, not learning biology.
Bullet 2. Naive predictors, which use zero biological data, are actually beating state-of-the-art neural networks on unseen drugs.
And bullet 3. Normalized metrics expose this memorization by subtracting the average, causing AI performance to tank.
Right.
So, question for you listening. If an AI is right for the wrong reasons, would you still trust it to make a medical decision for you?
Probably not.
Here's where it gets really interesting. If normalized metrics strip away that cheat code and prove the current state-of-the-art models are failing, we obviously need a better way to hold these algorithms accountable.
Before they ever influence patient care, absolutely.
Is that the specific gap the DrEval framework is trying to fill?
Yes, exactly. DrEval is designed to be a comprehensive, open-source benchmarking pipeline. It operates on a system called Nextflow.
Why is Nextflow important?
It's vital because it allows the entire evaluation pipeline to be executed reproducibly across different high-performance supercomputers. It adheres strictly to fairer standards.
Fairer standards. That's findable, accessible, interoperable, reusable, and e-reproducible, right?
You nailed it.
Which addresses the ongoing replication crisis in computational research, where scientists can't even reproduce their peers' results.
Exactly.
But how does DrEval handle the messy reality of the lab data itself? If the biological inputs fed into the models are flawed or noisy, standardizing the code won't fix the underlying issue.
That's where a crucial tool within the pipeline called Curve Curator comes in.
A curve curator.
Yeah. Historically, researchers might run 3 or 4 lab tests for a specific drug, get slightly varying dose-response curves, and just blindly average the results together to feed a single data point to the AI.
Creating a Frankenstein data point that never actually existed in the physical lab.
That's a great way to put it. Curve Curator normalizes the raw data, but it intelligently integrates the biological variability between those replicates.
So it doesn't just smooth it over.
Right. It treats the inevitable variance of lab work as a biological feature to be mathematically modeled, rather than just statistical noise.
So DrEval builds this highly rigorous, reproducible, perfectly fair arena, and then it invites the models in for a showdown. Who are the main contenders being tested?
Well, on the deep learning side, you had highly sophisticated architectures. Models like DIPK, which utilizes MolGet for encoding the intricate structure of drugs, and bionic embeddings for mapping the genetic networks.
Sounds incredibly complex.
Very. You also had models named MLI and SuperFeltar. These are designed to map out the most high-dimensional interactions between a chemical and a tumor.
And they were tested against simpler baseline models.
Yes. Baselines like SRMF, which relies on matrix factorization, Chamberta, which applies natural language processing techniques to read molecular structures as if they were text.
Using NLP for chemicals, interesting.
Yeah. And older classic machine learning algorithms like ElasticNet and Random Forest.
Okay, let's look at the LCO setting leaf cell line out. Since predicting for an unseen tumor profile is the core foundation of personalized medicine, when the grading rubric was finally fair and the data leakage loopholes were closed, who actually won?
In the LCO setting, none of the multi-million parameter deep neural networks significantly outperformed a properly tuned random forest.
A random forest. For those who aren't in the weeds of computer science, a random forest is essentially just a collection of simple decision trees voting on an outcome. It's a classic ensemble method.
It really is.
Why is an older, much simpler algorithm beating the absolute cutting edge of deep learning?
because of how the data is structured
Right.
and that pseudo-replication we discussed earlier. Deep neural networks are incredibly powerful, but they generally assume spatial or sequential relationships.
Like pixels forming an image or words forming a sentence.
Exactly. Tabular multi-omic data doesn't have that inherent structure. Furthermore, neural nets are so eager to reduce their error rate that they will find patterns in anything, including the random noise of a small, effective sample size.
But a random forest doesn't do that.
Right. By its very nature of ensembling multiple, shallow decision trees, it intrinsically performs feature selection. It's much more robust against overfitting the noisy, tabular data.
So the simpler model was better at isolating the true biological signal.
Precisely.
To figure out exactly what was distracting the deep learning models, the DrEval researchers used ablation studies, right?
Yeah, ablation studies.
That's where they systematically scramble different inputs to see what the model actually relies on.
Right. Think of it like systematically removing parts from a car engine one by one to see which specific component causes it to stall.
Okay, so what happened when they did that?
When they scrambled the basic gene expression data, the top-performing deep learning model, DIPK, failed. Its predictive power plummeted.
Which makes sense. Baseline gene expression is critical. But what happened when they scrambled the ultra-complex multi-omics? The copy number variations, the DNA methylation patterns that map out how genes are turned on and off.
When they scrambled the complex multi-omics, or even the high-dimensional molecular fingerprints of the drugs, the performance in the LPO and LCO settings barely registered a change.
Wait, if scrambling the fancy multi-omics doesn't change the outcome of the prediction, that means the AI wasn't even using them to make its decision.
Exactly. The features were mostly acting as useless mathematical noise.
That's crazy.
The advanced models were essentially looking at the baseline gene expression, combining it with the historical average potency of the drug, and completely ignoring the millions of data points of multi-omic profiling they were supposedly learning from.
Okay, third recap time. Bullet 1. The DrEval pipeline uses things like Nextflow and Curve Curator to finally create fair, standardized testing.
Bullet 2. Simple random forests actually beat complex deep learning models in personalized medicine settings.
And bullet 3. Ablation studies show those fancy multi-omics were basically just noise to the AI.
Which is pretty humbling.
Yeah. So listener, when was the last time you saw a simple solution completely outperform a highly complex, expensive one? Sometimes the fancy features are just hype.
That is very true. And if we connect this to the bigger picture, it gets even more concerning.
How so?
Well, everything we've discussed so far, the data splints, the ablation studies, the showdowns, has been evaluating models trained on lab-grown cancer cells against other lab-grown cancer cells.
Right.
The framework pushes this further with ex vivo generalization.
Ex vivo generalization. Right, because curing an immortalized cancer cell line that has been living in a flat plastic petri dish for 20 years is vastly different from curing a dynamic evolving tumor inside a human being.
Exactly.
How did they test this ex vivo leap?
First, they tested models trained on the CTRPV2 dataset against completely different laboratory datasets like GDSC1 and GDSC2. Even in this scenario, where you're still just predicting on cell lines, the model's performance drops significantly.
Wait, why? If it's still lab data, shouldn't the underlying biology translate?
You'd think, but it fails because of technical distribution shifts. For example, CTRP measures gene expression using RNA sequencing,
Okay.
while the GDSC datasets rely heavily on older microarray technology. The underlying formatting and distribution of the data changes, and the AI just struggles to adapt to the new context.
It's like studying for a multiple choice test and then being handed an essay exam. The formatting shift completely derails the model's pattern recognition.
It's a perfect way to describe it.
But the researchers went even further than just swapping lab databases, didn't they? They introduced actual patient tissue.
They did. This is the ultimate test. They applied these computational models to two highly relevant clinical datasets.
Which ones?
First, BEAT-AML2, which utilizes freshly isolated leukemia cells taken directly from acute myeloid leukemia patients.
So fresh cells, not immortalized lines.
Right. And second, PDX-Bruna, which uses patient-derived xenografts, specifically human breast cancer tissue that is physically grown in mice to maintain its complex three-dimensional architecture.
So we are moving from flat, immortal cell lines in a dish to highly complex, fresh patient tumors that have an actual microenvironment. How did the predictive models hold up when faced with actual clinical biology?
They failed dramatically.
Dramatically.
Yeah. The biological and microenvironmental differences between an immortalized cell line and living patient tissue, which interacts with immune cells, stroma, and a blood supply, are simply too vast.
The models just couldn't bridge the gap.
Not at all. It proves that taking a deep learning model trained on these massive public cell line databases and trying to seamlessly deploy it in a clinical ex vivo setting just isn't feasible with our current methodologies.
Okay, final recap for our listeners. We've got cross-dataset failure, meaning AI struggles when lab technology changes from RNA-seq to microarrays.
And ex vivo failure, meaning the AI collapses completely when tested on actual patient tissue, like the BEAT-AML2 or PDX Peruna datasets.
So, knowing all this, how should we balance our excitement for AI with the rigorous skepticism required for medical science?
It is a sobering conclusion for sure, but obviously a necessary one.
Right. So what does this all mean? Are we just hitting a wall with AI in medicine?
I don't think it's a wall. It's a necessary recalibration. Knowledge is only valuable when it is rigorously and honestly tested. This Nature Communications paper isn't saying that AI has no place in personalized medicine.
What is it saying then?
It's arguing that we are currently optimizing for the wrong things. We don't need deeper neural networks with billions of parameters right now. What we desperately need are larger, higher quality, standardized biological data sets.
And we need to actively eliminate pseudoreplication.
Absolutely. And most importantly, we need ungameable, transparent benchmarks like DrEval to hold researchers accountable.
We have to stop optimizing for the benchmark test and start optimizing for the biological reality.
Exactly. If we don't fix the foundation of how we evaluate these algorithms, we're just building faster engines for a car with no steering wheel.
That is perfectly said, and it leaves us with a critical thought to carry forward.
Yeah.
We've seen how Simpson's paradox, pseudoreplication, and data leakage fooled some of the smartest computational biologists in the world. It tricked them into believing an AI was brilliant when it was really just guessing the historical average.
It happens to the best of us.
It really does. So as AI rapidly integrates into our daily lives, from the autonomous systems driving our cars to the algorithms filtering our news to the tools screening our resumes, where else might these systems just be memorizing the average instead of truly understanding the complexities of the real world?
Are we optimizing for the test or for reality?
Exactly. When the systems you rely on make a decision, are they truly analyzing the problem, or are they just cheating on the rubric? Something to mull over. Thank you for joining us on this deep dive. Keep questioning the data, and we'll see you next time.